So I'm Chris Holman, CEO of Stratosvir. I wish I had a snappier title now, guys. I'm glad to read this, but we're engineering next generation viral immunotherapy to treat solid tumours and advanced cancers.
Say what it is.
Just a quick overview of the company. We're based in Stevenage. I could put a picture up there, but it's not quite as aesthetic as Cambridge. We're raising 650,000 to generate a data package to secure Series A funding. With a strong team, a lean organisation, and we've got the skills to take from the concept where we started to clinic. We've engineered a viral delivery system to treat cancer. It's underpinned by an antibody payload discovery platform, and that's all owned. The IP is owned by Stratosvir. We have in vitro and initial in vivo biodistribution data for proof of concept, and we're fundraising to extend this package for in vivo efficacy. So this is me. I've got, and my co-founder Antonio. This is the management team. Got a strong background in biologics discovery, actually starting with peptides, antibodies. My co-founder background in engineering viruses and cancer biology, and was supported by Priya Mande, who's ex-president and CEO of the Oncolytic virus company, Cioxis, but also deputy director of UK Vaccine Task Force, experienced in raising Series A funding and also partnership deals. So, you know, why did we start Stratosvir? Why did we think there was a need? Well, we were frustrated that sort of many cancer therapies provide sort of marginal survival benefit, but actually, you know, with a poor quality of life that goes alongside that. Immunotherapy, which is the current generation of cancer therapeutic drugs, is revolutionary. It's revolutionized cancer therapy. However, it's only relevant for the minority of patients. We want to make a drug that's relevant for most cancer patients, and we believe that viruses offer this potential, and it's the perfect immunotherapy agent that works in different levels with the immune system. It might seem like a crazy idea to inject viruses into you as a therapeutic, but there's a solid background behind this, in that tumours are the perfect breeding ground for viruses. You know, they have a sort of disabled immune response to viral infections, and also contain the building blocks for viral replication. So just a background to position where we are. So initially, back in the 2000s, 2010s, the viruses were used, but these were sort of attenuated viral vectors, sort of wild type attenuated, didn't really have any sort of immunotherapy payloads or potent immunotherapy payloads. Second generation viruses came along with antibody payloads and other sort of more exciting payloads. These are now playing out in the clinic, sort of late stage clinical trials, showing really positive data in sort of pancreatic cancer, ovarian cancer, bladder cancer, amongst others. But the problem with these two generations are that the therapy is rapidly eliminated if you inject them into the bloodstream, which is a problem. The workaround then is you have to directly inject them into a tumour, which is difficult because you need that surgical team, and it's only relevant for 20% of tumours. What we're doing at Stratosvir is engineering a stealth coat that avoids that initial sort of response to the viruses, so we can get the viruses into the tumour and we can potentially redose as well. We're also engineering the viruses so they can contain multiple immunotherapy payloads, which will lead to an increase in efficacy. So this is fairly generalist. It's maybe an overworked sort of metaphor, but I think it's relevant here, is that, you know, this is a Trojan horse approach. You know, we've engineered a stealth coat, so it goes under the radar of that initial response. It can get the virus into the tumour. The virus that we're using, vaccinia virus, does have a tropism for tumour vasculature. So the viruses enter the tumour, they infect cancer cells, they cause those, or they drive those cancer cells to make the therapeutic payloads that the virus carries. but also on another level, they kill those cancer cells directly. And this sort of releases tumor-associated antigens, neoantigens, and other markers that stimulate the immune response, open the doors to the tumor for immune cells to invade, and have multiple ways in which they can stoke up the immune system, patient's own immune system against the cancer. Also, viruses do modify tumors, so they can respond to other therapies, so they can work in combination with other therapies. So there's multiple mechanisms of action at work here that does have the potential to turn tumors that don't respond to immunotherapies, which are the cold immunologically cold tumors, hot again. One of our indications that we're really interested in, and particularly because prostate cancer is a sort of immune suppressed tumor, most people think that we've got a really good handle on prostate cancer, and we do. You know, men can sort of survive with the treatment, but for a few, and this is, you know, there's been a few examples, you know, with you know, famous celebrities here, that, you know, in certain instances where either you're diagnosed late or you have a particularly aggressive form of the cancer, you can rattle through the current standard of care, which are androgen receptor drugs and hormone therapy. And at that point, there are very few options. There are drugs that's coming through, such as radioligands and PARP inhibitors, but again, these don't make ideal drugs. And currently, immunotherapy doesn't work in prostate cancer because they're immunologically cold. But we see this as an incredible opportunity for our viral approach to really fire up that immune response within the cancers, turn the immune system on within those cancers so they do respond. And, you know, there's the rewards are there if we can transform cancer care, particularly in prostate cancer. We're looking at other indications as well, advanced liver cancer, hepatocellular carcinoma. But part of our business model as well is to out-license the viral vector, so pharma can use their proprietary therapies with, or deliver those therapies right into tumor using our viral vector. So this is how we're going to use funds. You know, 650,000. We'll be able to do in vivo efficacy studies, generate a data package that will help support a Series A fundraise and movement into the clinic. With this, there'll be a huge, we see a significant value inflection if we can get this data package, and also that's a data package that we can use to partner with pharma companies. We do have an Innovate UK grant for engineering antibodies, so that's the payload technology, our own proprietary payload technology that we use within the viruses as well. So we see it as a really good opportunity to invest in our company at an early stage and potentially a very high growth potential. We've got a differentiated type technology to treat solid tumors, and particularly where current immunotherapy checkpoint inhibition is failing. So thank you. I realise there's no data here as a generalist slide deck, but if you'd like to contact me, you know, we can talk through some data.
Chris, you do amazing work. Thank you so much.
All right,
yeah.
Thank you.
What we do, and with the limited funds that we have, and Chris is like plowing away.
The stealth code is the secret source. So even if it's non-energetic rodent or mouse preclinical model, have you got confidence it won't be non-energetic in human if it's a human? So systemic delivery goes wrong or very wrong. So have you done that? Homology, primates, what's your plan to get safety?
Yeah, it's complicated. It's different for different viruses. With vaccinia virus, our virus of choice, it's inactivated through slightly different mechanism. It's through the innate immune response. So it's complement inactivation.
Humans are 8%, neutrophils and mice are 14%, so you do get dramatically different.
Yeah, Yeah. So the immune system is different in mice and rats. We're doing the biodistribution in rats, which is more relevant to humans because of the difference in the complement.
How are you going to de-risk it first?
So vaccinia virus has been in the clinic, yeah, and for 20 years as an oncolytic virus, but also it's been in human population to eradicate smallpox. So this is the reason why we chose the virus as well, because there's a lot of clinical data with virus. So we don't think actually
That's not differential delivery. So your stealth coat is going to be a different engine.
the stealth code. Well, okay, I have to get into the. I have to get really deep into the biology of bacterial virus, but we can chat sort of later if that's possible.
You're going for late stage solid tumours, so you might expect metastases as well. Do you expect that your technology is going to be just as effective on the metastatic cancers as
Yeah, exactly. Okay. I think with the recent release of the neoantigens and that immune response, educating the immune system, that should have abscopal effects for those metastases as well.
The question I was going to ask was the abscopal effect question, right? Because as everyone knows, in the heady days of radiotherapy, this is the thing. So if you can demonstrate the abscopal effect, that's what everyone in big pharma wants, right? So if you can demonstrate that even in a mouse, that I think would be a huge value because all the guys, that's what everybody wants, right? Because cancer's coming late, but if you can stop the prostate cancer spreading to other parts of the body, that's a massive differentiator, which I think many of the other approaches just don't.
Yeah, if we can go in early as well with the disease, that would be fantastic.
Thank you so much. If anyone's interested, reach out to me or to Chris, and we can discuss. It's a small round basically to get them over through the pivotal data. So I think it's a good time.